Okay, this is the pH of Household Solutions lab. All right, the first thing you have to do for this lab is turn to the data table, and in the first column you need to record all the different solutions that are being used in this lab. So we have a list right here, so go ahead and fill out your first column. Baking soda, ammonia, Gatorade, water, mouthwash, vinegar, and a mystery solution. So fill out the whole first column. You might have an extra row or you might have to add a row depending on what copy of the lab that you have. Press pause if you need to finish filling out that column. Okay, the next thing you have to do is you have to fill out the second column. You're going to make a hypothesis if those solutions are acidic, basic, or neutral. So finish filling out the second column. For the mystery solution, you'll truly have to just take a wild guess. Alright, so what we've done is we've taken each one of the 7 solutions and we've put a sample of it in the well plate. So here's a diagram that corresponds to what's inside of each one of these wells. We're going to run some pH indicator tests on each of the solutions. We're going to run a red litmus paper test, a blue litmus paper test, a pH paper test, and a cabbage juice indicator test for each one of them. So let's go across the table now and let's just run all the tests on our first solution on baking soda. So this is a piece of red litmus paper, and it does look pink, but they do call it red litmus paper. I'm going to dip that in the baking soda, and now what you're going to do is you're going to record the color that the paper has changed to under the red litmus paper column. The second test, so this is the 4th column in your lab, is the blue litmus paper. I'm going to dip that into the baking soda. Notice that the color has remained the same. You're not going to write same, you're not going to write no change, you're going to write blue. Going on to the next column, it's the cabbage juice. So I'm gonna add a few drops of cabbage juice to this. Notice the original color of the cabbage juice is a blue color, but I'm going to drop it into here, which is the baking soda, and you're going to record the color. Our last test is going to be the pH paper test. Looks similar to litmus paper, except it's a different color to start with. I'm going to dip that into the solution, and how pH paper works is we then compare that color change to a chart on the side of the vial. So you estimate what pH number you think this color is representing. Okay? So those are all the tests for baking soda. So now on the data table, what you need to do is in the final column, you need to answer this question. Based on all your results from the litmus paper, the cabbage juice, and the pH paper, is this solution, the baking soda, an acid, a base, or neutral? We're now going to repeat all those tests on the remaining 6 solutions. So our next solution is ammonia. This is the red litmus paper. Sometimes you'll notice that it takes a while for the paper to change. So I'm going to let that just sit there for a moment. I'm going to move on to the blue litmus paper. So I'm going to dip the blue litmus paper in there. Okay, so record the color of the blue litmus paper. You can kind of see that we do have, if I hold it up to the white paper, it might be a little bit more obvious, but we do have a color change. So record that color change. Again, this is for ammonia. We're moving on to the cabbage juice. I'm going to put a few drops of the cabbage juice in here. Record the color. And then finally we have our pH paper. And we're going to hold that up to the chart, and you make an estimate of what pH number you think this color best matches. Okay. We're going to move on to our next solution, which is the Gatorade. So this is the red litmus paper test in Gatorade. This is the blue litmus paper test in Gatorade. This is the cabbage juice indicator in Gatorade. And this is the pH paper. This is the indicator. Okay, and remember we have to match this to the chart. So take a look at the numbers, take a look at the color of the paper, and you select what pH you think best matches. Okay? All right. Moving on to our next solution down here. This is water. This is tap water taken right out of the faucet. So we have red litmus paper. We're dipping the red litmus paper in. Note what color it is. This is the blue litmus paper. Make a note of what color. We're going to put the cabbage juice into the water. Make a note of what color. And finally we have our pH paper. We're going to hold that up here, show you the different numbers, and you decide what number you think it best matches. This is for water. Okay? Moving on to our next solution here, it's the mouthwash. Here is the red litmus paper test. Here is the blue litmus paper test. Here is the cabbage juice indicator in the mouthwash. And then our final indicator test is with the pH paper. So again, make your best estimate what pH you think the mouthwash is. Okay. Moving on to our next solution, which is vinegar. This is the red litmus paper test. This is the blue litmus paper test. This is the cabbage juice indicator. And here is the pH paper. All right, moving on to our last solution, which is the mystery solution. We have the red litmus paper test. We have the blue litmus paper test. We have the cabbage juice test. And we have the pH paper test. So don't forget, in the final column, based on all your data, you're writing if the solution is an acid, a base, or neutral. When you're all done with your data table, there are several questions on the last page to answer using your data. That is the pH of Household Solutions Lab.